Okay, now we're going to continue on with photosynthesis, picking up where we left off Monday. So again, on Monday, we kind of left off talking about photosystem 2 and talking about the pigment molecules that are contained therein, between 200 and 500 chlorophyll pigment molecules. These are attached to 3 to 8 polypeptides. Now, the energy from light that is then trapped or collected by these chlorophyll molecules is going to eventually be transferred to the photosystem 2 reaction center via resonance energy transfer. So essentially how this works is that, imagine here we have light, kind of signified by this lightning bolt, is going to excite the electrons on this chlorophyll, illustrated by this disk. That's going to transfer the energy to the next chlorophyll, and it transfers to the next chlorophyll, and then to another random chlorophyll, and that energy is going to keep bouncing around among these chlorophylls until eventually it's transferred to this reaction center, this together, which is the photosystem 2 reaction center. We're going to come back to how exactly that works in just a few minutes. So the reaction center itself consists of about 20 polypeptide chains. These are coming from chloroplast DNA, but it also contains a special chlorophyll molecule known as chlorophyll P680. It's called 680 because it absorbs light predominantly, or peaks light absorbance is at 680 nanometers. Then the energy from the light, the energy from the light harvesting complex. The light harvesting complex being all the other chlorophyll molecules that are in the reaction center are going to transfer their energy to the P680 chlorophyll. And this is going to raise an electron in the P680 to a higher orbital. And then that electron is going to be transferred to Plastoquinone, which is the next element in the electron transport chain here. So again, light is going to excite electrons on one of these chlorophylls in the light harvesting complex, and then essentially that energy is going to be passed between chlorophylls somewhat randomly until eventually it makes its way to the P680 chlorophyll molecule, which is then going to pass it on to the primary electron acceptor in the reaction center, which then passes it on to Plastoquinone. It's supposed to be a P, not an R. There we go. Okay, but once this happens, now that final chlorophyll, the P680, is missing an electron because it passed it on. It's now sitting on Plastoquinone and heading down the electron transport chain. So how does that get replaced? It actually turns out that there's another protein in the reaction center here called the oxygen evolving protein. It has 3 polypeptides from the nuclear DNA. It also contains manganese, a chlorine or a chloride, and calcium ions in it. And this particular protein is going to split water. And it's going to transfer the electrons that are generated thereby to the P680, regenerating the proteins. Now, one of the regenerating or refreshing the electrons, in the P680. The interesting thing here is that then you have this oxygen that then is going to be free, and then it's going to bind with another oxygen to make O2, and this is where we get the oxygen that actually photosynthesis generates, is from this reaction here. So again, this is being simulated right here. Here is the reaction center of photosystem 2. We're going to have water then that is then split, making a proton, a couple protons, and then also an oxygen. This oxygen is then going to be leaving the plant or whatever photosynthetic organism this is. And the electrons, essentially from water, are going to be then transferred along the electron transport chain. Now, it's kind of after the fact, but these are going to be replacing the electrons off the P680, but then those become the electrons in the next cycle that will then be donated and so on down the line. So really the electrons for this electron transport chain are coming from this water. Okay, so let's begin to talk about the next element in this electron transport chain, plastoquinone. So plastoquinone is a lipid molecule. It can transfer 2 electrons at a time. So right now, this should be sounding a lot like something that we talked about earlier on in the mitochondrial electron transport chain, particularly coenzyme Q. And again, that element is right here in this diagram. And this is sitting on the stromal side of the membrane. So not on the thylakoid lumen, but on the stromal side. And because it's a lipid, this is really very mobile. And this is the thylakoid membrane is very fluid, and this is a hydrophobic lipid, so plastoquinone can travel through this membrane very, very quickly, and it makes it very, makes it very convenient for passing electrons to the next element in the electron transport chain. Okay, and that next element here is cytochrome B6F. Now in this case, the cytochrome B6F in the photosynthetic electron transport chain is very similar to the mitochondrial BC1 cytochrome that we talked about earlier. And it is sitting right here, B6F, and is going to transfer across, it is a 4 polypeptide chain, one of them coming from the nuclear DNA, and 3 of those polypeptide chains coming from the chloroplast DNA. It also contains an iron on heme groups and is an iron sulfide polypeptide. An electron is going to transfer an electron from plastoquinone to plastocyanin, and in the course of doing that is going to be pumping some protons across. Each one electron is going to lead to 2 protons being pumped from the stroma into the lumen of the thylakoid. And therefore, we're actually passing a pair, so we get a total of 4 protons per water that we split being pumped across. Then it passes on to plastocyanin. Plastocyanin now can only transfer 1 electron at a time. So that is sitting right here. And it's going to transfer between cytochrome B6F to photosystem 1. So plastocyanin, this right here, cyanin, literally stands for kind of a bluish color, but often is given to molecules that contain copper. And the copper is going to be the metal that's helping carry the electron. So without the electron, the copper is 3+, but with the addition of the electron, it becomes copper 2+ to carry that electron. Now, plastocyanin is interesting. It's somewhat similar to some other of the elements, particularly cytochrome C, that we have been seeing in previous electron transport chains. It's not bound to the membrane. Instead, it's free in the thylakoid lumen. Now, so it's actually even more free than the cytochrome C that we had talked about before. But plastocyanin is just not bound to the membrane. It's instead just free in the thylakoid lumen. This makes it very mobile, again, kind of like plastoquinone, and makes it very able to transfer electrons between the cytochrome B6F and onto the next element. And so what it is transferring electrons from is cytochrome B6F to the photosystem 1 reaction center. And like I said just a moment ago, it's really, really very mobile. Again, really kind of making this very similar to the cytochrome C that we talked about in the mitochondrial electron transport chain. Okay, now that brings us to photosystem 1. And photosystem 1 is in this diagram sitting right here, and it's essentially going to receive light energy and re-energize the electrons that it's holding at any given time. So like photosystem 2 that we talked about, it has a light harvesting complex, and the photoreaction center that the light harvesting complex has 11 polypeptide chains. 6 of these are from nuclear DNA, and 5 of these are from chloroplast DNA. And the photoreaction center is going to contain another chlorophyll P700. So if you remember from the previous reaction center that had P680, P700 is a chlorophyll that has a maximal absorption at 700 nanometers. So slightly different, slightly modified from the chlorophyll that we were seeing in the previous reaction center. So the light energy that photosystem 1 then gathers is going to cause an electron to reach a higher orbital like we saw before. And that electron is going to be transferred to Ferredoxin that we're going to talk about after this. So really, photosystem 1 is essentially re-energizing the electrons that are traveling past the electron transport chain at this point. Okay, so let's go into talking a little bit about Ferredoxin. Ferredoxin is going to take one electron at a time, and it's going to pass it on between that to the NADPH reductase. So that is sitting right here. So ferredoxin is on the stromal side of the thylakoid membrane and is an iron-sulfur protein. It also, like some other elements that we've seen before, is very mobile because it is sitting kind of just, it's a peripheral protein, it's not embedded into the membrane, and it transfers an electron to the NADP plus reductase from the photosystem 1. So let's then talk about NADP plus reductase. That is sitting here, which is also sometimes called the ferredoxin NADP plus reductase, hence the acronym here FNR. And this is going to transfer our electron to NADP plus, which is the final electron acceptor for our their photosystem electron transport chain. Now, I want you to kind of notice how essentially this entire electron transport chain in photosynthesis has acted in essentially an opposite manner than what has happened in our mitochondrial electron transport chain. So remember, in the mitochondrial electron transport chain, the initial electron donor in that chain is an NADH, which then passes off, so another nicotinamide adenine dinucleotide, and it passes off an electron to the chain, which passes it along, pumps some protons across the membrane, and at the very end donates those electrons to oxygen, which is the final electron acceptor, and that oxygen goes on to form water. In the case of the photosynthesis electron transport chain, instead what we're doing is we're taking our electrons from water, passing them along the electron transport chain, and then the final electron acceptor is another nicotinamide adenine dinucleotide. So kind of exactly what we saw in the mitochondrial electron transport chain, but in reverse. And once this NADP plus accepts the electrons, is going to become NADPH, and we have 2 electrons that are held per NADPH. Okay, now we've built up all of these protons. It's time for us to actually do something with the proton motive force that we've built up to this point. And we're going to use that essentially the exact same way that the mitochondrial electron transport chain did. We're going to use it to generate ATP. And we're doing that with an ATP synthase. In this case, the CF0-CF1 ATP synthase. And that in this diagram here is sitting here. Again, we are using our proton motive force here and creating ATP from ADP. So per electron pair, we had, and remember, per electron pair that was donated from water, we had 2 protons that were created at the oxygen evolving complex. That, remember, is from water. We took those essentially directly from water. And then we had 4 protons that were pumped at the cytochrome B6F. Now, a proton gradient has been set up, and thus a pH gradient. The pH is 8 in the stroma. Again, this is, remember, outside the thylakoid membrane. And on the inside, in the thylakoid lumen, we are at 5. So again, remember, this is actually a larger gradient than what we were seeing in the mitochondria. There's actually a 1,000-fold difference in proton concentration, whereas in the mitochondria, we only had an 8 to a 7 pH change, only a 10-fold difference. So this is quite a lot larger proton gradient than what we saw before. However, it's electrically neutral across here for various reasons that I'm not really going to delve too deep in, but it's essentially electrically neutral, which means that we have a proton motive force of about 200 millivolts. That's not all that much more than the 120 millivolts that we had across the inner membrane in the mitochondrial inner membrane, but it is still greater, and it's all due to the pH gradient. We do not have an electrical gradient in this case. So, 4 or 5, a mean of 4.67 protons will pass through the ATP synthase per every ATP that we end up creating. And this process is known as photophosphorylation. This is in contrast to the oxidative phosphorylation that we had in the mitochondria. Okay, so we have now used light to build up a proton gradient. We use that proton gradient to phosphorylate ADP and make ATP. So we've essentially trapped the energy and convert it into a biologically useful form. What do the plants end up doing with that now? And the answer is that they are going to use that in the Calvin cycle, which is something happening in the stroma of the chloroplast. It is also known as the C3 pathway. This was discovered in 1945. It earned Melvin Calvin the Nobel Prize in 1961. So, in the case of what is essentially happening here, CO2 is being fixed. And it's going to be incorporated into organic molecules. Eventually, it's going to be incorporated into everything from carbohydrates, amino acids, fatty acids, and nucleic acids. But the very initial thing that's going to be incorporated into is carbohydrates. And all of this is going to be powered by the ATP and the NADPH that we created during the photosynthesis electron transport chain, or during photophosphorylation. So, let's talk in general how this is working. We're going to get incorporation of CO2 into an organic molecule. Essentially, we are going to take a 5-carbon molecule, add a 1-carbon molecule to it, which is the CO2, and we are going to get 2 3-carbon molecules. So we've taken an organic molecule here, the 5-carbon molecule, an inorganic molecule, the CO2, and get 2 organic molecules out of it. This is how we get, therefore, this integrated into organic molecules. The specific molecules involved here are going to be ribulose 1,5-bisphosphate, which is the 5-carbon molecule, plus CO2, is going to yield us 2 of the molecule 3-phosphoglycerate, or 3-PGA. And this is why the Calvin cycle is called the C3 pathway, because it ends up creating this 3-carbon molecule for the plant. Now let's talk a little bit about the enzyme that is responsible for this, and that is ribulose 1,5-bisphosphate carboxylase oxygenase, which is also known by a shortened term, RuBisco, which sounds like just a delightful name. Ribulose bisphosphate carboxylase oxygenase. And RuBisco is a heterodimer, has 1 polypeptide from nuclear DNA, and the other polypeptide is coming from the chloroplast DNA. Now, RuBisco itself is kind of a slow enzyme. It only catalyzes 3 molecules per second, which in the grand scheme of enzymes is not very fast. However, what it lacks in speed, it actually makes up for in abundance. There is a ton of it in the thylakoid membrane, or excuse me, not the thylakoid membrane, but in the stroma. A lot of it. It's 50% of the chloroplast protein is RuBisco. And RuBisco is the most abundant protein in the world. There are a lot of plants out there, and a lot of them are performing photosynthesis. And in each of those, half of the protein is, well, half of the chloroplast protein is RuBisco, which means that there is a whole bunch of this protein on Earth. There's about 20 pounds of RuBisco in the world for every person on Earth. That is a lot, especially for one single enzyme. Okay, so CO2 fixation. that actual reaction does not require an input of energy. You might be thinking, whoa, you just told me we're going to use ATP. Yes, that's right, but we don't use it in the step that fixes the CO2. We'll come back to exactly how that works. At the end of the day, the Calvin cycle is going to release glyceraldehyde 3-phosphate, or G3P, and it's making that, again, using ATP and NADPH. The glyceraldehyde 3-phosphate is then moved into the cytoplasm using phosphate cotransport, and is used to make other organic molecules. We can use the glyceraldehyde 3-phosphate to make a plethora of other things, and sucrose is the main one that these plants, or most plants, are going to be making with it, which is the main transport carbohydrate. Then we need to regenerate the ribulose bisphosphate from the other glyceraldehyde 3-phosphate that we had. This is what requires the ATP. It's not the actual carbon fixation that does it, not the step that Rubisco catalyzes. It is the step of regenerating all those products back to reactants again. It's going to require the ATP. So essentially what we have here is we have 3 carbon dioxide plus 3 ribulose bisphosphate plus 9 ATP and 6 NADPH is going to give you 3 ribulose bisphosphate again. So we've regenerated that. We've regenerated that by using up those 9 ATP and these 6 NADPH. At the end of the day, we have our glyceraldehyde 3-phosphate, which is what we want to, which is kind of our fruit at the end of the day. Now, this, you see, is regenerated, so we can now, once we're at this point, start this again. We just need to get some more CO2, which we get from just in the atmosphere. The plant needs to get some more NADPH and ATP, which it gets from the photophosphorylation, and it can run this whole thing again and just keep producing the glyceraldehyde 3-phosphate essentially indefinitely, as long as it has the light and the CO2 to do it. Thus, per every carbon that we're integrating, so divide this all by 3, we are going to use 3 ATP and 2 NADPH to regenerate this ribulose, these 3, or each ribulose bisphosphate. Okay, so let's talk about the specifics of this. So again, we're going to start with a ribulose 1,5 bisphosphate and a carbon dioxide. Through the action of Rubisco here, we are going to get 2 3-phosphoglycerate. So that's what we got here, 2 3-phosphoglycerate. Each of these 3-phosphoglycerate, then, we are going to get, through an intermediate step here, a glyceraldehyde 3-phosphate. So we have taken both of these 3-phosphoglycerate that Rubisco has made, we make them into 2 glyceraldehyde 3-phosphate. From this point on, one of these glyceraldehyde 3-phosphate is going to become ribulose 5-phosphate again. We're going to regenerate the ribulose 1,5 bisphosphate, and eventually back all the way to the ribulose 1,5 bisphosphate that we're going to be using. The other glyceraldehyde 3-phosphate that we make is going to be exported out of the chloroplast back into the cytosol, where it can be used to make other organic molecules. I already talked about that. Okay, so that is the normal course of photosynthesis. We have gone from light, and then we've gotten ourselves all the way to an organic molecule made of carbon dioxide, where we're good to go. We have taken energy, we've trapped it into this storage organic molecule form, which is eventually sucrose on the inside of the plant. However, this wonderful happy story is about to turn sour, because we are going to talk about photorespiration, which is kind of the dark underside of photosynthesis. Photorespiration is a biological flaw. It does not have any redeeming features to it. It is simply a biological flaw in Rubisco that all photosynthetic organisms have to deal with. It's a complete waste of about 20 to 50% of all plant carbon, ATP, and NADPH. And this happens for no reason. It happens for no good beneficial reason to the plant. There is a reason for it. The reason for it is that the Rubisco active site that you use to react carbon dioxide also reacts with oxygen. Pretty much just as well. In fact, slightly better for oxygen, in fact. And so, if carbon dioxide concentration is low and oxygen concentration is high, instead Rubisco is going to catalyze this reaction, where we get ribulose, take a ribulose bisphosphate, and then oxygen. Instead of a CO2, we get 1,3-phosphoglycerate, which we can use, but then we also get this thing called a phosphoglycolate. So essentially, we've gotten back here, this 3-phosphoglycerate can then be turned into a glyceraldehyde 3-phosphate, and that glyceraldehyde 3-phosphate we can generate back into a ribulose bisphosphate. So we're good there. But the problem is, this phosphoglycolate is gone. This is bad. Like this, you do not want phosphoglycolate. This is so bad. So essentially what we've done is we've taken a 5-carbon molecule, a 0-carbon molecule, which is the oxygen here. We've taken a 3-carbon molecule, which is useful, and then the phosphoglycolate, which is a 2-carbon molecule, which has to be gotten rid of and recycled. And essentially, this is lost to us. At least part of that is lost to us, which means we're losing some of our carbon here. It's not good. So the 3-phosphoglycerate can still be used. That we're okay with. It gets essentially turned back into the 1,5-ribulose bisphosphate. Now the 5-phosphoglycolate, however, has to be recycled, and it's expensive to do this. It's not without costs. Number one, the phosphoglycolate itself is toxic. This, if it builds up too much, could kill the chloroplast. It's also going to inhibit glycolysis, specifically at the enzyme triose phosphate isomerase. This is also not good. You want to be able to use glycolysis. That's fundamental. And because of this, because of how this works, because you get the phosphoglycolate, which is a 2-carbon molecule, sometimes this is referred to as a C2 cycle. So the phosphoglycolate is going to be recycled. This is going to involve peroxisomes, which we're going to talk about later on in the quarter, and also mitochondria. It's a slow process, and it's energetically expensive. And we're doing this in close proximity to photosynthetic tissue. In short, what's going to happen here is we're going to take 2 phosphoglycolates. We're going to turn them in the peroxisome. We are going to turn them into glycine. So we have to take the phosphoglycolate. We're going to transport it to the peroxisome. And the peroxisome is going to be turned into glycine. These 2 glycines together can then be used to make a serine. We're losing a CO2 here. This is actually why we call it photorespiration. We're using light. It is going to use an oxygen and release a carbon dioxide. So again, we are just losing that carbon dioxide. And then this serine, then through a series of steps in the peroxisome and some input of ATP, becomes a 3-PGA again. Not great. So we started off with 4 carbons here because we have 2 carbons in this phosphoglycolate, 2 carbons in this phosphoglycolate, and we end up with a 3-carbon molecule having lost our CO2. Blech. Not good. In the end, is that we're throwing away 3 ATP in this course. 2 NADPH that were used in the first place. Those that we use in the process of doing this, they're gone. And and this is this is what was used to fix that CO2 that we lost in the first place. We used these 3 ATP and these 2 NADPH to fix the CO2 that we lost, and now we just lost that. We're losing the carbon dioxide, and also so this is just really a waste because we're losing lots of energy that we used to get that carbon dioxide in the first place. So most plants are going to lose somewhere between 20 to 50% of all their fixed carbon dioxide via photorespiration. This is not the ideal situation. Nothing really wants to do this. In hot and dry environments, though, plants need to keep their stromata closed, which are essentially the openings on the other side of the leaves in which they can bring in and exchange gas with their environment. They have to keep these closed to avoid water loss. And this leads to a problem because you have photosynthesis going on. Photosynthesis is using up carbon dioxide and it is creating oxygen. So the carbon dioxide is going down, the oxygen is going up, and this is increasing the probability that Rubisco is going to use oxygen as a substrate instead of CO2, which favors photorespiration. Again, this is a biological flaw that is not helping us out at all. So, if CO2 is high, we have 2 Rubisco, 2 CO2, and we're going to get 4 3-PGA. If you have low CO2, we're going to use 2 Rubisco and 2 oxygen instead, and we're going to get 3 3-PGA and 1 CO2, which is now gone. Goodbye. You've just lost all the energy that you used to get. Now, some plants, especially ones that are in water limiting or hot situations, have found a way to not completely eliminate photorespiration, but to at least ameliorate it. They use a different enzyme. So some plants, C4 plants, which we're going to talk about why they're called C4 plants in just a little bit, use a different enzyme to fix CO2 at low carbon dioxide levels. These C4 plants include things like corn and sugarcane and crabgrass and dandelions and a handful of other things that are really useful to us. And what's going to happen is instead of using a CO2 plus ribulose 1,5-bisphosphate, you're going to take CO2 plus a phosphoenol pyruvate and turn this into oxaloacetate. It's called the C4 pathway since we're fixing carbon dioxide into a 4-carbon molecule instead of a 3-carbon molecule. The enzyme that does this is known as PEPCO, which is phosphoenol pyruvate carboxylase. So phosphoenol pyruvate. This is RuVAT. Carboxylase. And it has a very high affinity for CO2. Therefore, it can bind CO2 even at low CO2 levels. The one problem with this is that it takes additional NADPH and ATP used. It's not quite as energetically efficient as Rubisco is. So it makes it more energetically expensive to fix carbon dioxide. However, that being said, it's not nearly as expensive as losing up to half of your carbon by photorespiration. So, I must have looped back. Okay. So, really, at the end of the day, the C4 pathway, we have this biochemical adaptation using the PEPCO to actually, or Pep-C to do this, but it will allow, it's also an anatomical adaptation that's going to allow high CO2 in certain parts of the plant, particularly where we're doing using Rubisco. So it doesn't actually totally cut out Rubisco. I'm going to show you a diagram of how this works in a few moments to show you how that works. But essentially what we can do is in the mesophyll cells, where we're going to have more contact with the atmosphere, we're having our C4 enzymes. And CO2 can be low. However, in the bundle sheath cells, we'll have our C3 enzymes. And in this case, we can keep CO2 high. One of the ways that we keep CO2 high in those areas is that we have no photosystem 1, or no photosystem 2, only photosystem 1. So no oxygen is produced in the bundle sheath cells. We're keeping oxygen low in those places, CO2 high. So this is essentially how this works. In the mesophyll cells, we have Pep-C or PEPCO, depending on which one you use. Phosphoenol pyruvate here is going to take CO2 or when it's an aqueous, bicarbonate, and we're going to turn it into oxaloacetate. Oxaloacetate in the chloroplast here is going to be turned into malate. So here turned into malate. That malate then is going to get transferred to the bundle sheath cells, and that malate then, through work of another enzyme, is going to be turned back into pyruvate, which is going to release a CO2. Now that we have CO2 in the bundle sheath cells, we're going to use that CO2 in the Calvin cycle, all the while keeping the CO2 high and the oxygen low, so Calvin cycle works without Rubisco undergoing photorespiration. So essentially this is a way of concentrating CO2 in the bundle sheath cells and doing our work of actually initial fixation of the carbon dioxide with Pep C out here, where oxygen is going to be higher and CO2 is going to be lower. Kind of give you an idea of how this works in an actual plant. This is a cross section of a corn leaf, and here we have stomata down here. They're opening up and allow oxygen and CO2 into the cell, actually more oxygen out. And here in the mesophyll cells, all these green ones here, we're going to be having the C4 enzymes in there. So we're going to have Pep C actually converting the CO2 into oxaloacetate, and then eventually into malate. That malate is going to be taken into the bundle sheath cells. So those are these purple ones here, and you see how they are protected from the atmosphere. So we can kind of control the gases that are here. And we're going to be taking that malate there, converting it back into CO2, so we're keeping the CO2 high. Because photosystem 1 is not present here, or excuse me, photosystem 2 is not present here, remember that has the oxygen evolving complex, we are not going to be using them to build up additional oxygen. So CO2 will be high, oxygen will be low in the bundle sheath cells, and we'll be able to run we'll be able to run Calvin cycle, and specifically Rubisco, without, or minimizing at least, photorespiration happening. So that is C4 photosynthesis. Basically, it's acting as a way to trap carbon dioxide and pass it on to the C3 pathway. Okay, that's not the only way that plants do this. There's also another one called CAM photosynthesis. CAM stands for crassulacean acid metabolism. Really, this comes from comes from a genus of plants called Crassula, which are kind of these fleshy, succulent plants. But it was first discovered in these plants, and this is a particular type of photosynthesis that happens. We first found them, and it's been called crassulacean acid metabolism. And essentially, this is a way to again separate CO2 uptake and the C3 enzymes. Instead of doing this in space like the C4 pathway did, we're going to separate it in time. So again, C4 pathway was separating them in space, where we had the fixation of carbon dioxide happening in the mesophyll cells, and we had the C3 enzymes working in the bundle sheath cells. CAM, on the other hand, is going to separate these out in time rather than space. So let's take a look at how this works. During the night, when the temperature is low, the plants will open up their stomata and allow CO2 in. We're going to take that CO2, and using Pep C again, we're going to turn the CO2 into oxaloacetate, and again into malate. So we're still at this place. We're turning that into malate, and then that malate we are going to store in a vacuole. And as we do this, we're going to keep the CO2 low in here. We can keep essentially having it diffuse into here and just essentially concentrating malate. Then, and it's actually going to be then turned into malic acid, which is a little bit easier than malate to store in aqueous solution in the cell. But then during the day, when the sun is hot and we do not want to be losing water out of these stomata, the plant will close its stomata. At this point, to keep CO2 high in the cell, and we cannot bring it in from the atmosphere. Instead, we are going to take malic acid, turn back into malate, ship the malate back out, and we're going to run this backwards, back to oxaloacetate, and back to CO2. That's going to go into the Calvin cycle. So now we can keep the CO2 high inside inside the photosynthetic cells without opening up the stomata and losing air, or excuse me, losing water. Now, again, this is essentially just the same thing that happened during C4 metabolism, but instead of this taking place in two separate cells, like it happened in C4, we're having it two different times. Essentially, during the night, when temperatures are lower, we can accumulate CO2 and have our stromata open. During the day, when temperatures are high and we don't want to lose that water, we can then run this whole thing backwards and release that CO2, feed it into the Calvin cycle. Okay, that pretty much finishes up photosynthesis, and next time we are going to pick up with some protein production, but that is it for photosynthesis.